Hi all, and welcome to the lecture video for Module 2.2 of Unit 1. As always, you can see the learning outcomes here and use these to help facilitate your study in addition to the study guide. Again, this module is pretty much a review of your genetics section of Bio 1. So we're not going to dive too deep into this because most of this information should be merely a review for you. I'll just cover some of the highlights here and hit some of the points that may or may not be on the exam. So the macrostructure of DNA is basically you have strands of DNA that are supported by proteins that are called histones, which help to wind the DNA around those proteins. And this helps to condense the DNA into what's known as chromatin, which is then packed tightly into chromosomes when the cell is ready to divide so that all that genetic information can fit inside of the nucleus. Protein synthesis is very important, and if you refer back to the cell organelle tables, you can see where this takes place in the cell. Basically, in the steps of protein synthesis, you have a few different things. You have transcription and translation. In transcription, this occurs in the nucleus. Basically, the DNA template strand is used to make complement RNA strands. RNA is then modified to form mRNA or messenger RNA. That mRNA leaves the nucleus through the nuclear pores and then needs to be translated into a readable form. So this occurs in the cytoplasm in a process called translation. Go figure. rRNA or ribosomal RNA binds to the messenger RNA and reads the codons or the triplet base pairs of DNA. And then transfer RNA transfers the appropriate amino acid, which is the monomer functional group of proteins, to the ribosome, and then proofreads it using its anticodon, or the reverse triplet. Amino acids are then covalently bonded together by peptide bonds to form a polypeptide, and that complex polypeptide then takes on its primary, secondary, and tertiary, and eventually quaternary levels to form those complex protein structures that we spoke about in the last module. There's a few different ways that RNA is processed. One is splicing of DNA. So in the nucleus, a structure called a spliceosome cuts the introns out. We're not going to focus too much on this. Just know that this is how some of the information is removed and processed out of RNA. So as far as the genetic code is concerned, DNA holds all of the genetic information necessary to build the cell's proteins, which then carry out the functions that the cell needs. So the nucleotide sequence of a gene is ultimately translated into an amino acid sequence of the gene's corresponding proteins. And what we're talking about is that codon is that triplet base pairing that helps to code for a particular amino acid. If you ever take biochemistry, you'll learn all that stuff, and it'll be way too in-depth. You'll have to memorize all of them. But you'll start out with a DNA strand. So if you have a particular strand of DNA here, you should all remember what the base pairs of DNA are, your A, T, G, and C, and how those pair up. You have T that corresponds with A, and C that corresponds with G, and vice versa. We talked about earlier how those nucleotide structures are formed and how those interact and bond together. In the process of transcription, it takes that DNA template strand and makes it into a complementary RNA strand. When I'm talking about questioning here, I would strongly encourage you to focus on whether I'm asking you for the complement RNA or the complement DNA strand, because that affects the type of nitrogenous base that you're going to use in there. So if I'm asking for DNA, you're going to see A pair with T and G pair with C. If I'm asking for RNA, you're going to see T pair with A pair with U. From there on out, that RNA strand is going to be translated and code for a particular protein or amino acid. This just rehashes what I was just talking about. So if you can tell here, if we start with this triplet template, this is going to be the DNA that codes for that. this is going to be the RNA that codes for that. I'm not going to dive too deep into the cell cycle just because, again, this is just a refresher from Bio 1. We also cover this in lab. But that DNA replication is necessary to distribute genetic information into daughter cells so that cells can continue to divide. The idea of semi-conservative DNA essentially means that one strand of the DNA is copied and then introduced to the parent strand. So when you make new copies of DNA, you're going to have one side of that strand as the parent strand or the old copy, and then one side is that new copy. So it's semi-conservative. Conservative meaning that it would retain both parent strains, semi meaning that one parent strain and one new strand. This is about as deep into mitosis as I'm going to go into the lecture portion here. I would strongly encourage you to take a look at this chart as some of the unit questions come directly from it. As you're looking at this, it's also going to be important that you take note of the microscopy that you're going to see here, or what particularly is going on in the cell at the particular time and the particular phase that of cell division that the cell is in. While you're in lab, it's going to be imperative that you're able to recognize, based off of a microscope slide, which phase of cell division that cell is in. So in addition to knowing what happens, for example, in anaphase, the centromeres are going to split in two. Sister chromatids, which are now called chromosomes, are pulled apart towards opposite poles or opposite ends of the cell. And then the spindle fibers begin to elongate in the cell, meaning that they stretch out and connect. So in addition to knowing that, you're also going to be going to need to be able to recognize that this is what that looks like in comparison to, say, prophase here. Interphase is going to be the long process of the cell division cycle. This you may not have covered so much in Bio 1, but it's essentially your G1 or your G2, which are your gap phases, and then your S phase, which is your synthesis phase, which essentially is the period where cell DNA replicates itself. And this is where DNA also checks in on itself. So because DNA is replicating during these phases, you're going to have a lot of checks for errors in that DNA replication process. And as the cell is doing that, the body can develop cancer. Cancer cells are body cells that have lost their ability to regulate cell division. And this usually occurs with the uncontrolled replication of DNA, which results in the uncontrolled replication of cells themselves. So cancer cells will form from what's known as a tumor. The development of cancer from a single abnormal cell into a collection of cells or a collection of abnormal cells known as a tumor. You may have heard this if you've heard anything about cancer before, but there are two types of tumors, the benign tumor and the malignant tumor. Benign cancers are still tumors. Make note of that. But they have not yet invaded the surrounding tissue, meaning that it's cancerous, but it just hasn't traveled to another part of the body. Malignant cancers are the inverse, in that tumor cells have begun to invade the surrounding tissue. We can also consider this as a mastitization of that tumor. Make no mistake, the benign tumors, although they have not metastasized and moved to different areas of the body, can still be dangerous and cause problems. A lot of times in pop culture or in today's idea, we have the incorrect notion that if a tumor is benign, it cannot harm us, when in fact it still can.